So let's take a look at the specific events which occur in meiosis I. As I said before, meiosis consists of 2 rounds of cellular divisions. Probably the most important events occur during meiosis I. This is where we get crossing over, this is where we get independent assortment, and this is where we go from having a diploid parent cell to having 2 haploid cells that eventually will be giving rise through another round of division to 4 daughter cells total. So let's take a look at what happens in meiosis I. The key events, the things you absolutely must know about meiosis I: we reduce the chromosomal count from 2 complete sets of chromosomes that we start out with in the parent cell to having 2 cells each with only 1 complete set of chromosomes. So if we were talking about humans, we'd start out with a precursor cell that had 46 chromosomes in it, and after meiosis I, we would have 2 cells each with 23 chromosomes in it. Okay, we're going from diploid to haploid. We also have 2 other critical events happening in meiosis I that I briefly mentioned already, and those are crossing over and independent assortment. Crossing over occurs in prophase I. Independent assortment occurs in mitosis I, and the purpose of both of these is to generate greater genetic diversity. You'll be happy to know that with both meiosis I and meiosis II, we have the same predictable sequence of events that we had in mitosis, i.e. we've got prophase, prometaphase, metaphase, anaphase, telophase. We put a number designation after each of those to indicate whether they're happening during the first round of meiosis or during the second round of meiosis. Last thing, and this is another key point to remember about meiosis I. During meiosis, we are separating, homologous chromosomes, not sister chromatids. Very important point. We are separating homologous chromosomes, and because we're doing that, that allows us to go from having 2 complete sets of chromosomes inside the cell to having 2 cells each with 1 complete set. So let's see how this happens. Here we start out in prophase I. And guess what? All the events that you learned about that take place during regular prophase of mitosis also occur in prophase I of meiosis I. So all the events we discussed before, like the nuclear envelope disappears, the nucleolus disappears, the centrioles begin to move apart from one another and grow spindle fibers, the chromatin condenses into visible chromosomes. All those events occur during prophase I, and also during prophase I, we get that crossing over occur, which is what you can see happening here in this cell, crossing over. Okay, we go into prometaphase I, and this is where, remember, the centrioles have moved to opposite sides of the poles of the cell. They've grown out their spindle fibers, and the spindle fibers have connected onto the kinetochores. We move into metaphase I, and in metaphase I, our homologous pairs line up as pairs. Notice that we don't have something like this where we have single chromosomes lining up along the middle like we do in mitosis. Instead, in metaphase I of meiosis I, we get homologous pairs lining up along the metaphase plate. Big difference there. And that is where we get our second major important event occurring, which is that independent assortment, where the chromosome from grandpa may be on one side and the chromosome from grandma on the other, or the reverse could be true. This helps to mix up the genetic components. Then we move into anaphase I, and you will remember this is where we pull apart. But now, instead of pulling apart sister chromatids, we pull apart homologous pairs. So you will notice here in this picture that I still have a fully duplicated chromosome moving to each end of the cell. Okay, so notice if we count it up here, we now have 2 cells that are 1N each, and I'll count it up for you. Let's go back to the previous slide. You can see in this picture here I have 1, 2, 3, 4 chromosomes total. 2 homologous pairs of chromosomes for a total of 4 duplicated chromosomes. When I pull apart those homologous pairs, I have 1, 2, 3, 4 chromosomes now, but they're divided between what is about to become 2 separate cells. So I have gone from 2N to 1N in each cell. Okay, that's anaphase. In telophase, all the same events happen in regular telophase. You do the opposite of everything that happened in prophase. So we just reform the nuclear membrane, we reform the nucleolus, the chromosomes may start to become uncondensed, etc., etc. The centrioles start to diminish their spindle fibers, etc. And then we reach a point where we sit kind of in a I don't know, sort of a limbo zone for a little while. This is called interkinesis, and it's sort of a waylay point. We're not quite done yet. We did go through, by the way, cytokinesis 1 here. You can see because we have separated into 2 separate cells, cytokinesis 1. All right, now we're going to go through the second round of meiosis, called meiosis II.